Novartis to try and set up NASH. So I'm not a believer in working in NASH because it's very difficult.
Right.
Brian,
Well, yes, I mean, we did actually briefly discuss this over the last couple of days, and I think the feeling of the group was that it's a difficult disease, there are no good animal models, and it requires attacks at multiple points, so not just anti-fibrotic agents, that's the sort of end stage. It's all too late then. You really need to tackle also the metabolic alterations that are driving the whole process of fatty liver and degeneration. So treatment of obesity and insulin resistance, type 2 diabetes, and these sort of things are very important in preventing NASH. I think that was sort of the conclusion we came to.
Yeah, that's absolutely true. Most type 2 diabetics, especially if they're obese, develop NASH, and so there's an insulin resistance basis to the disease. And then, you know, the fibrosis is it's not consistent. It's and the animal models are not. They far better reflect the diabetes aspects of the earlier disease, but not of the more end-stage fibrosis, and it's a big issue.
Right. But listening to you gentlemen, it seems like fibrotic diseases in general have this problem. They're so complicated. We're waiting for the basic science to be worked out before the pharma companies would get involved. John, would you concur with that view?
Yeah, I mean, I think we've got to take into account within pharma in general, there's been a change in what size of disease you get into. In the beginning, you know, it was completely market driven. We have to have large populations, and that's what we look at. But another way of doing drug discovery and development is to actually work out, do you know the molecular mechanisms that's involved? Right. And that'll improve your chances of being successful in whatever drug you develop. That's why companies got into cystic fibrosis. It's a great example of a sort of monogenetic disease. So you know what your target is. It's either modifying CFTR, either potentiating or correcting it like Vertex did and we've done, or you look for other ways of hydrating the mucus by causing things to stimulate chloride release from the epithelial cells. So you can get into smaller diseases. It's much easier if it's single genes involved because you know what you're going for, right, or something that does the same thing. But when you get into somewhere in between, you've got to take some bets. And I think NASH was a bet we weren't prepared to take. We just thought it was too complicated.
I mean, there are a few companies. There's quite a few companies who are investing in NASH. At Receptos, because we had a basis around G protein coupled receptors, we took a look at the GLP-1 receptor and its role in type 2 diabetes and did it play a role in NASH. And there was a study done with liraglutide not too long ago that was published, a small study, suggesting that liraglutide, which is a type 2 diabetes drug, had some effect on NASH patients in terms of reducing the rate of fibrosis development. So we actually started a program at Receptos. We ended up identifying something called a positive allosteric modulator that had a dramatic effect on weight loss in animals. It lowered blood glucose, and when we did do some of these NASH models, which are basically leptin genetically deficient animal models that are fed high-fat diets, you can see some fibrosis. We saw dramatic effects in fibrosis there. That small molecule the IND has just been filed, so we'll see where it goes. So there is, it's interesting. We saw a discussion about liraglutide in chronic kidney disease. Sometimes you find serendipitous observations in clinical trials that are relevant to another disease, and maybe that's what's happening with a drug like liraglutide.
Right. Yeah, I think they're difficult.
In that point, do you think that's why big pharma are interested in fibrosis, is that there's a lot of crossover? We've heard that today.
Yeah, I think so. Like the SGLT2 inhibitor, liraglutide. Yeah. I think that's the case. But you're absolutely right. I mean, we know so much more about the pathology of an animal model than we do about human disease, and
we looked at it with some of our tyrosine kinase inhibitors, and designing trials is very difficult in that indication. Because they're going to be fairly long term. Even the proof of concept studies are going to be probably a year or longer, and it's difficult to know what the endpoint is. If we had some better biomarkers, I think people might be encouraged to go into NASH and to look for therapies. But I think we need to be able to see acute effects that in the long term will pan out by positively influencing the disease process. So we need to have circulating biomarkers of collagen production, and it's a balance of new collagen formation and breakdown which occurs as well. So you need to be able to measure both processes. But there is a group actually in Novartis that's looking at this at the moment and has generated some antibodies that will identify neoepitopes on collagen that are circulating and things like that. So that's what we decided to do, not to do anything in NASH, but actually to try and go after identifying some biomarkers.
All right, so we've got biomarkers improving. Robert's talking about the models improving. Now, the model you were describing is a very recent one, if I understand.
Yeah.
Like no more than 5 years old.
No.
Right. So it seems like the tools have only just become available to go after this area.
Yes and no. I think a lot of the fibrosis diseases, because it's such a long-term disease and you really don't want to be doing trials for 2 years, a lot of effort is actually going into finding biomarkers which reflect the ongoing disease and are much easier to measure, and you could actually conduct a trial for a much shorter period of time if you can cause that biomarker to go up or to go down, if you believe that's reflecting the ongoing disease and what you've done. But this is it's still a rather a problem in a lot of the fibrotic diseases.
You're left with biopsies. I mean, that basically was the outcome, and then they're not very good biopsies because you can hit a spot where you get little fibrosis or another spot where you've got a lot. It doesn't give you the overall picture.
You heard from she hears, I was not here, and the lung clinician, this typical thing in the CT scan is honeycombing. Well, this honeycombing isn't all the way through the lung. It's very patchy.
Yeah.
So again, if you're driving the thing just based on biopsies, you've got to make sure you're when you take your biopsy, you're in a fibrotic area.
Right.
Yeah.
Now, so
Well, I was just going to say, at Receptos we licensed an antibody from AbbVie called, it was an anti-IL-13 antibody. And the biology of IL-13 suggests it has both pro-inflammatory biology as well as pro-fibrotic biology. We decided to take that antibody, it was already phase 2 ready, we took it straight into a disease called eosinophilic esophagitis, which is a massive accumulation of eosinophils in the esophagus. Patients have trouble swallowing, there's wasting, and there's significant tissue remodeling, fibrotic activity going on in the esophagus. So we took it into a phase 2 trial, and we tried to put as much science into the trial. We tried to understand whether the drug was working or not. But we had reasonable endpoints, although some of them are patient-reported outcomes, which are tough to variable, right? But we tried to, biopsies was part of the diagnosis and part of understanding whether the drug worked. So we got tissue, which was good, in multiple parts of the esophagus.
We did exactly the same.
And we made sure the biopsies were deep enough so that we could look to see if we're having an impact on fibrotic pathways as well. The whole idea here was to try and develop the drug in eosinophilic esophagitis, get as much information as we could from our clinical trial, and said, well, look, if we're having pretty significant anti-fibrotic effects, let's think about IPF, or let's think about, life cycle management, right? Take your drug into your primary indication, and if you see something going on that's pretty significant, like anti-fibrotic, then why wouldn't you try something like IPF?
right. Now,
We did exactly the same. Yes, we had our own anti-IL-13 and went into a severe asthma trial in a proof of concept, and it showed that if you well define your patients and select out the right ones prior to the study, you had a very good effect. We also did eosinophilic esophagitis. It works in that as well. We also did IPF. Didn't do a damn thing.
so go back in time. The IL-13 and asthma, where did that program end up in the end?
Well, in Novartis, there's various reasons why a thing continues or not. Usually it's not just based on pure science and clinical outcome. There has to be the right personalities around and the right champions. And actually IL-13 was a target that a lot of companies went for. Glaxo, Astra, there's 3 of them now in phase 3 trials for asthma. I think Novartis is currently trying to out-license their anti-IL-13 right now.
Right. Now, when I was preparing my note the other day, I noticed a certain amount of drop-off in terms of development area within the IPF space. I guess there's a lot of, in pharma land, a lot of people are thinking, well, we've got 2 drugs, they slow the rate of decline, let's forget about it.
No, well, not from my experience and the people I've talked to in and out of Novartis, and we talk to lots of other people now that I'm out of Novartis, is basically there's still a large unmet medical need, there's no doubt. I mean, pirfenidone, the number of trials they tried to do to show success was over about 10 years. And there's still a large body of physicians believe pirfenidone doesn't do anything.
Right.
The Boehringer compound came out of their oncology program. It's one of these multiple tyrosine kinase inhibitors, and you're not quite sure which one is the important tyrosine kinase. But if you look at the actual trials that, again, the physician put up there, the dropout rate was quite amazing. It's a very high dropout rate in those trials with the tyrosine kinase inhibitor.
No wonder the nausea and the gastrointestinal
Yes.
upset.
Yeah.
Right.
What you've actually got, Stuart, is a roadmap for companies now to get a drug registered in IPF, given
Right.
what pirfenidone.
Yes, yeah.
And that helps a hell of a lot
Yeah.
because there's FDA accepted endpoints now for registration, and that actually attracts companies into the space.
So let's talk about that situation. Transitioning back to NASH. Obviously, no one's ever got this far before. So the agency is still finding its way. What can we expect in the next 5 years? Say if Intercept works out with what they're doing in terms of what the agency is going to be asking for.
They want to see resolution of non-alcoholic fatty liver disease, and they want to see no worsening of fibrosis. They're the 2 major clinical endpoints that I'm aware of.
Yeah.
Right.
For
Right. You've just seen them halve the rate of patients for the phase 3 that they've gone into. So it feels to me like it's still a work in progress for the agency, what they're looking for in terms of their studies. Have you gentlemen seen that before, where there's a new area where the agency is still feeling their way, so the endpoints change over time?
Oh yeah.
Well, yeah, that happens all the time. I mean,
It's a
if you go
movie.
back to simple sort of respiratory diseases like mild asthma, with all the bronchodilators that were developed by GSK,
Yeah, that I'm
Astra,
sure.
and myself. Basically, all they were interested in was FEV1, post expiratory volume, and 1 second we talked about earlier. But as you look at different mechanisms of actions in those diseases, anti-inflammatory, anti-fibrotic, FEV1 is not what your readout needs to be. It needs to be a whole range of different readouts which actually reflect what's going on. So, yes, I think you've got to be prepared if you go into a new disease to have constant interaction with the regulatory authorities and actually persuade them that the actual endpoints that are out there right now may not be the appropriate endpoints that you will have in the future.
And it's tough to validate a biomarker or a patient-reported outcome, or it's tough to get those, right?
Yeah.
In eosinophilic esophagitis, there's still significant discussion about what patient-reported outcomes should be considered registrable.
Yeah.
So we've moved into the era where routinely, if there's something hot, that big pharma will acquire the relevant developer, be it privately held startup or in some cases public. How does that change the culture of a company like, say, Novartis, as they realise they have to acquire more to stay ahead of the curve?
Well, as I was saying, I think that we look at a whole range of diseases all the time, but the 2 most important things that came out the way we looked at things was, is there an unmet medical need, a real unmet medical need? So we're not really in the range of Me Too's, which a lot of people were. And in that disease, do you know what's going on?
Right.
And unless if you don't understand the molecular mechanism going on, what's the point of entering it? You're just going to be like throwing dice.
Right.
So that's how we selected. It wasn't based on market size, it was based on, do you understand the molecular mechanism? And is there a real unmet medical need? And that's the one we will target. And no matter how big the disease is, you can sort out your pricing to deal with that later on.
Right.
I think that's also pharmacoeconomic elements have to be measured in future if you want to get a price. And with so many biologics now around commanding, ever increasing prices,
Right.
people are very sensitive to this. So that in designing phase 3 studies, it will be increasingly necessary to show that by giving your new drug, you're actually not increasing healthcare costs, but rather, there are benefits, there are economic benefits of using your drug. so that not only have there to be efficacy and safety endpoints, there will also in future be increasingly a requirement to show.
This wasn't complicated enough.
Yeah.
Sorry, Stuart, does that have big implications for combination therapy and your strategy as a company? So if you think, fibrosis is a disease with multiple pathways, and they're likely to be additive or synergistic effects of using multiple therapies, but clearly there's one payer and who wants one outcome. Is it something that you need to be thinking about controlling all the assets in a combination as soon as possible in order to get something to market? Or is that likely to come, those kind of combination trials are likely to come phase 4?
I think so. I mean, up till now, there are some examples of where a combination has been developed from the outset, but usually the regulators want to see efficacy of the two components separately before you actually do the combination studies. So they're more likely to occur sort of post-registration. But the ever-increasing cost, because there are several of these biologics, say, in an area like maybe fibrosis in the future, I think we're going to be expected to deliver also, you know, these pharmacoeconomic parameters to show that, you know, you give the drug, you stop hospital admissions or concomitant therapy or whatever it is.
It's pretty tough, I think, to get two large pharmas together, each with a registered drug, with the notion that you could get the two drugs together and have an additive or synergistic effects.
It does happen.
It does happen, but it's not that common.
It's a tortuous process.
It is, yeah.
Because I certainly know, I've certainly seen in oncology people hoovering up assets in development because they're predicting that that's going to be an area where there's going to be a lot more combinations in the future.
Right.
I'm just wondering if fibrosis, because of the pathology of the disease, may end up looking a bit similar to that.
I'm convinced it will. It's a very complex disease.
Yeah.
And the other thing is, because that's why I asked about standard of care in one of the presentations. Now we have a standard of care in IPF, pirfenidone, and I can't remember Nintedanib. Well, not in Australia, but at least in Europe and in the US. It'd be very difficult to do a trial where you weren't looking at your agent on top of standard of care. You couldn't do it just by itself. I would think that would be quite difficult these days.
Not when you've got mortality outcomes like you saw.
Yeah.
I mean, when mortality's significantly impacted, you wouldn't be able to do a placebo-controlled trial over a longer period anyway. So
would it be worth trying to do one in a market like Australia so that you could do a head-to-head? Be ethical in Australia.
That might be a reason for doing the trial where you were able to do a trial without the standard of care. And you weren't just putting it on top. Yeah.
Hell, you don't even need GMP material for phase 1 trial.
we discovered
this yesterday, so this is a lot of an eye opener.
That's great. It's fantastic.
Speaking to Glenn, I think as a clinician, he would be unlikely to give a drug. He would give the standard of care, still Innate and Nintedanib, and anything would be on top of that.
Yeah. That's what would happen in Europe or the US. Yeah.
But coming back to your original question about combinations, you know, there are some examples where companies will go after combinations, particularly in the oncology area. So,
Novartis went out of their way to acquire a company just because they wanted that one to be a combination in a certain cancer treatment.
Yeah.
But is it in the end that you then, in terms of costs, you've got like drug A, drug B,
Yeah.
and you can't actually price it like A plus B, you actually have to price A plus a little bit. And that's about it, isn't that where we're heading? And that's why companies will want these combinations because the payers, the restrictions that they put in place, that you really have to have this portfolio that you give to them, that you actually get on the list and where people want to go.
Well, there's ways to answer that.
The generics might help because as drugs go off patent, it could get cheaper for combination therapy.
But you're thinking of like bundling drugs?
Bundling or particularly in oncology where everyone has now whatever you want to choose, and then you go. I think we're going to see it. I think in diabetes we probably may see it play out now with Lantus and all those where how they're really Sanofi's price it quite differently, where it's not A plus B anymore. There's that whole move from the payers, which I think for fibrosis is very important because it is the massive expansion in capital spending.
Yeah.
So it's just would like to get your input. How do you see that play out in the end? How are we going to do save costs?
Yeah, I'm not sure that I can really comment from any experience there. on that one, although...
Well, there are diseases where that's happening already. Again, going back to bronchodilators, you know, you start off with your long-acting LABA, which is 24-hour plus, then you've got your long-acting LAMA, which is 25-hour plus. But most people do fixed-dose combinations of the two. But now there's triples being developed, where you have the long-acting beta-2, the long-acting muscarinic, and the steroid, all within the same inhaled device. So it depends on what disease you're talking about, because it's actually much easier if everything's in the same blister pack, all right? But I think it's difficult to predict in the diseases we've been talking around here about what will be the combinations, because we haven't got anything that's really effective yet. So.
Do you then, I guess, a totally different question, but on going back to NASH, where you've got some companies, I think Gilead, for example, trying to go into late stage NASH and say, well, shorter trials, maybe we can affect something more here, maybe it will immediately impact hospitalisation. Is that a way to kind of tackle the disease slowly and move forward?
We were given the opposite advice. Enroll patients that had it goes F1, F2, F3, F4. Get the F1, F2 patients. Don't get the late stage
But
ones.
do you get them easily? Are they presenting easily?
So I actually don't know the answer to that.
Yes, I did a bit of work on that with a company called Resonance Health. Basically, I mean, NASH isn't something that shows up for a long time. There's 30% of the population has fatty liver disease, and
Right.
the 3 to 5% will show up. But the doctors are now getting smart about the need to go look for those people.
Yeah.
And in the case of Resonance, they've got at least a easy to use MRI for fatty liver. So I think the day is coming when we'll pick up patients a lot earlier, and it won't be in the too distant future.
And the other question is whether you can, if you go for end-stage disease, whether you can find things that would reverse or pre-existing fibrosis. And there are a couple of trials now being done, I think in IPF, with agents where they claim at least there is the opportunity to reverse already pre-existing fibrosis.
Yeah.
That would be...
That's huge. I mean, if you can show that, not just delay progression, but actually reverse it.
But I'd get you into a completely different other area. Instead of just inhibiting the fibrosis, you want to be stimulating the regeneration, where you're actually getting new alveoli formed and things like that, because that's your problem. Your alveolar disappears. The same in COPD, your alveolar disappear. So all the things that we've been talking about is basically slowing down the progression and trying to normalise. But it's something that people are beginning to talk about, but there's nobody with any compounds or even targets as yet. There's some targets where you actually stimulate regeneration of the airways.
Off the top of your head, how many centres around the world are specialising in lung fibrosis mechanism?
Are you talking about pharma, academia, or
Academia.
Oh,
Yeah,
lots. Most major countries have the realisation that fibrosis. Well, my area is in the lung end, IPF. You could name any European country and various US cities, and in Australia. I mean, there's a lot. There's no.
So what questions have they been answering for the last 10 years then?
Remember, for those centres to do a trial, they need the compounds or the antibodies, right? And these things have to be. It's only recently that people have been looking at new targets within this area. The pirfenidone, if you look at the history of that goes back years and years. It's sort of about it must be about 12 years or so, I think, when they started looking at pirfenidone. But I'm not quite clear what your question is, what you're asking.
One of the major things that they're asking was the question today about the type of cell that causes the disease and where it comes from.
Yeah.
There's a lot of work on that worldwide, and we still don't know.
There's still a big argument about, is it epithelial cells transforming to fibroblasts? Is it endothelial cells transforming to fibroblasts? There's this big story that came up, Bob Streeter was the main pusher of this when he was at Michigan and LA, but there's quite a few other groups that have taken the story up. These things called fibrocytes. So fibrocytes aren't fibroblasts, and they're not monocytes. They have markers of each, but they seem to be particularly good at turning into myofibroblasts. And at least in the IPF field, most people believe that myofibroblasts are quite important in terms of the collagen deposition and contributing to the pathology. But it's by no means 100% agreed that that's the thing you should be targeting.
John, staying with you for a moment, you give, I learned this from Mesoblast here, you give patients mesenchymal stem cells, they'll traffic preferentially to the lungs. So regenerative medicine, as you said, is going to be a big area
Yeah,
in your
there's certain
field.
little biotechs have already set up and are going that, and there's a lot of academic activity, but it's still a long way to go.
Right. Given that the first of the regenerative medicines are on the cusp of approval, that it's likely that would be a big R&D activity for your old
Yes,
employer.
there's most big pharma are looking at that now,
Right.
and smaller ones, yes.
Right.
But but don't let me give you the impression that this is just round the corner. It is not. It's a hell of a long way to go.
Yeah, I think the mesenchymal stem cells, if they're given IV, they land up in the lung because
Yes.
that's the first capillary bed they get
Right.
to. They
Right.
get trapped there,
Right.
So
One of the biggest problems, though, is what they end up being.
Right.
And do they end up being what you want
Or
or
just
something
pump out
not?
growth
And that
factors,
still hasn't not quite fully
and they go back
understood.
to the bone marrow as well.
Yeah.
Yes.
But would you agree that in large pharma, when they see a small company, a small biotech doing something pretty interesting, like in this field, rather than trying to reinvent the wheel
No,
in-house,
it's much cheaper for them to just acquire it.
that's right,
Yeah.
and the skills that come
The
with
skill
it,
sets
the
and everything
technology,
with it,
yeah.
Yeah.
And that happened in this area, didn't it, with the compounds you were looking at, the LPA1 receptor.
Oh yeah, Mira. I mean,
Were
that's
acquired,
a
yeah.
great example. Mira was acquired by BMS.
BMS,
BMS wanted to be in fibrosis,
Yeah.
and they had the pretty good at that time LPA
One and two.
one
How big was that
receptor.
transaction when it happened?
Oh, I can't remember.
It was quite big.
It was 1.2 billion, I think.
Yeah.
Yeah.
Brian, let's go back to you. Gilenya came up a couple of times in the conversation.
Yes.
Somewhat controversial drug, shall we say?
That's a great drug.
It's a great drug.
It makes Novartis an awful lot of money now.
Roberts is going to be slightly better, but it still makes a billion plus.
Yeah, absolutely. I mean, a big step
is a
forward in its time. Take us back in time. You discover that it's got these cardiovascular issues. Is there a no shit moment within the development team saying, what the heck do we do now? That you're then able to subsequently overcome as the program moves forward?
Well, the problem was, as I said, we didn't have a preclinical model that would predict the bradycardia because the animals don't show. There's no species, including even monkeys, that would show this effect. So one had to go into the clinic. We could have theories about, maybe it's not the receptor that you're needing to get the trapping of the lymphocytes in the lymph nodes, which is the effect that you want. That it's, one of the other sphingosine receptors that it's hitting.
There's
that's
5
responsible.
sphingosine receptors.
Yeah.
Gilenya hits 4 of them.
Right.
Roberts hits 2 of them.
Right.
Basically, his one doesn't hit the one that's probably driving the fibrosis or the bronchoconstriction, which is the CYP3.
Right.
But that's actually opened up a whole new area.
Yeah.
If you can come up with a CYP3 antagonist, you're going to treat airway hyperactivity.
I mean, Gilenya was originally developed. I remember when the first papers came out. I read about
Mitsubishi.
it. Yeah, I was quite
Pharma.
intrigued way back then with it.
Yeah.
But it was originally developed in Novartis for solid organ transplantation,
Yeah,
wasn't it?
yeah.
Absolutely.
And they spent a number of
It
years.
came into my group,
Yeah.
actually.
Yeah.
But we identified the MS indication based on a model that we developed of a modified EAE model. It was actually, which is an animal model of multiple sclerosis, and we had a chronic model. So EAE is, as it says, it's acute allergic encephalitis, and we developed a model that had more of the characteristics of the sort of relapsing and remitting type of disease where Gilenya and your compound is now successfully used. And we identified it as being something that worked well there. We looked at, as you have done, at many other autoimmune diseases as well. But we had to go into the clinic before we found this effect on the heart rate.
Right.
And it seems to occur with the first dose. We don't really understand that either. So after you've
The
had the first
subsequent
dose,
dose, it doesn't happen.
subsequently you don't have the same problems.
Right. So transitioning this over to what we're talking about in the fibrosis field. There's a lot of drugs which we might find out there are problems as the first of those drugs hit the streets that could not reasonably be foreseen beforehand. That's a reasonable assumption.
That's true with any disease.
Yes, I think with antibodies the chances that you get an off-target effect are of course a lot lower. Most of the toxicities are related to hitting
this is the beauty
something
of
else.
this technology, right? Before people didn't think antibodies could be used to target GPCRs or ion channels, which is usually the domain of the medicinal chemists with the low molecular weight compounds. But if you then look at the failures with low molecular weights, particularly with ion channels, there's desperate need to find another technology that does it. And
Right.
these small i-body things seem to be able to do that.
Right. So stick with you for a second, John. You've had a number of monoclonals in your career.
Yes.
Talk to us about some of the lessons you learned as you were working in
Well, you see, in the
that field.
beginning, antibodies were used to target structures or proteins or whatever that weren't accessible to low molecular weight compounds. It was either too big a receptor-ligand interaction. But now, with things like the i-bodies, you can actually go for any surface structure like a GPCR or an ion channel, which allows you to do that. But in the past, all the antibodies that were out there, I mean, Novartis is a great example. Xolair, the anti-IgE, which we did with it was with Genentech. If that came out today, it probably wouldn't get registered because it's not actually, it's quite a low affinity interaction, and it doesn't actually bring down the IgE levels as low as you want them to. But it does still work in lots of people in terms of severe asthma. So that got us into sort of looking at antibodies, but most of the antibodies were targeting soluble proteins that were floating around. IL-13, IL-1, IL-33. That's still conventional monoclonals can still do that quite effectively, and if they're humanized, you reduce your risk of immunogenicity. But this technology is another league again.
Okay, so looking in from the outside, if you hadn't got involved in this company in a more professional capacity, what would you see as the risks to the i-body platform?
Okay, I think they have from their CXCR4 data, they've basically shown they can do it with GPCRs. We don't need to get into which ion channels, but there's some preliminary data with some other ion channels they've got into that says these can work on that as well. Right. And if they can demonstrate the real selectivity and specificity that low molecular weight compounds can't do, they'll be basically unsuccessful. It opens up a phenomenal area.
Right. I think the two questions always when you go with a new protein into man is, are you going to have problems of immunogenicity?
Right.
Yeah.
You know?
Right.
And is it going to hang around long enough, for example?
Yeah, well, you've cracked
to
that
have an
puzzle now.
effect?
So buy some more stock. The stock's going to be 6% share.
No,
But it looks like...
But yeah, go on. Sorry.
Yeah, I was saying, in terms of specificity, and particularly with these agents, to get at things that other antibodies can't, that's the great advantage.
There's a huge, there's not that many great targets out there. There really isn't. I mean, when you look through the whole industry, there's not that many targets. There's a big bucket of what you'd call undruggable targets. Right. And that includes a lot of GPCRs and ion channels that are known to be associated with pathology in different diseases. I think AdAlta's got some technology that allows undruggable targets now to be drugged.
Right.
Yes.
That's what I think. So.
Yeah, I was effectively saying it a slightly different way, but it's the same.
Let's drag Mick into this. So, Mick, why are we spending more time on intracellular targets here? These i-bodies are pretty small, and most of the undrugged...
No, you still have a difficulty with that. It's still, you won't get it in the cell, but
Right.
Yeah.
And they've got disulfide that would probably be ripped open in the reducing environment itself. So there's enough targets on the outside.
Yeah, there is.
Let's leave those alone for a while. All right, so how much time we got, Sam?
I guess 5 minutes, 5 or 10 minutes. Does anyone else have any questions?
Okay. Yeah. So Robert told a lovely story about a company that grew and raised more capital in his seed rounds and venture rounds than probably accounts for 25% of the venture capital we have totally in Australia, and then went to an IPO and raised $700 million in a year. You know,
You're lucky.
we're in Australia. We simply don't have access to that sort of money. Should we just focus on what we're good at, proof of concept, be it early stage drug development, target validation, identify some leads, and then pack up shop and go to the US? Or is it worth ploughing away through the...
I would not pack up shop and go to the US. I think there's some unique characteristics about being able to do research here in Australia. For me, so I understand being how cash strapped a lot of companies are here. What I'd try and do is try and attract US capital in That's what I'd do.
Absolutely.
And that's through either VCs or through crossover funds or public. There's well-heeled public funds over there, and what you've got to do is tell them why you're a value proposition to them.
Right.
And that's just what every other American company does. This is an incredibly exciting industry, and getting it to a certain point and then giving it off to America is for me is not the way to go. I think what Australia needs is successes. Where you can take something pretty novel through to clinical proof of concept somewhere.
Yeah.
You'll be amazed. You'll attract capital. I'll guarantee it. Right now it's a cash-strapped environment. That's tough, and you guys are basically skeleton crews. I mean, we were lucky. We could go out and hire 20 people and move things incredibly quickly. You don't have that luxury, but you're doing things in a focused manner that is advancing these programs pretty well, despite those limitations. And I'd keep plugging away, and I'd also entertain talking to funds in the States. Attract money here.
Right.
A good drug.
You can't talk to people in the US.
A good drug program here is as good as a good drug program. For me, a good drug program here is as good as a good drug program
There's also Europe,
in
I
America.
think everyone always says
Yeah.
go to the US, I think Europe.
Yeah, there's Europe.
Yeah.
a lot of- yeah.
Yeah, I can't really speak for Europe. We didn't even bother to go to Europe for raising money.
You didn't need to. No.
No.
Well, what's your view of when did companies need to go through that US IND process? I know you're on the Innate board, and they haven't done the IND,
Yeah.
and at Receptos you got the IND straight over to all through US channels. What's your view on that?
About getting an IND.
About
and I'll-
what the appropriate time to go through that US IND
if
process?
you can do some sort of clinical trial here in Australia first, and then take that information with you to file an IND, you're almost guaranteed success.
Yeah.
Any kind of clinical data
You've
trumps.
got clinical data, yeah.
But
But I mean, your IND is normally submitted after you've got the results of your toxicology
Tox
studies.
GMP talks.
That's what really will
GMP talks.
will trigger it. Once you've got those data,
Yeah.
then you need to get down to writing. Took him 10 months.
no sleep.
Well, Robert, what you did was phenomenal.
Yeah.
I never believed that those were the timelines. That's fantastic.
Yeah.
We never achieved anything like that in the labs.
Well, we could because we had nothing else to do.
Right. Please think up panels. Oh, hold on.
One
we've
more
got-
question. Why did Novartis not license or do partnership with Receptos?
Well-
Knowing the
Yeah.
advantages of their compound. They may have spoken to you, I know, but
Yeah, we spoke to everybody, but I think they had a lot of confidence in Gilenya. I think they're thinking about extending the patent life of Gilenya, and there's different ways of doing that. They got a backup molecule called saponamide, which has gone into secondary progressive MS.
Yeah, you had it there.
Yeah,
BAF.
Yeah, BAF312, yeah. So,
They had a big program,
Yeah.
essentially, but
yeah.
We had a
Big
big
chemistry.
internal program.
There was a lot of other
against all
targets they were
CYP
looking at.
receptors.
A lot of other targets they were looking at,
so it,
I mean, we were surprised with Celgene, to be honest. They weren't an MS company.
yeah,
They-
but isn't it, well, I guess the rumour brought through it always been that they only required it because of the other parts to it.
The IBD?
MS, yeah.
That's a vicious rumour, that one. They had a strong interest in inflammatory autoimmune diseases, and they wanted to expand their franchise into other indications, and they wanted to be opportunistic as well. They realised a cornerstone that they've got a strong interest in inflammatory bowel diseases. They've got a good infrastructure there, and so they saw that clearly as their lead. But they also saw MS as a leg into expanding their autoimmune inflammatory empire. Yeah.
Please thank our panelists. Yeah. Thanks, guys. So, we've got plenty of time. Yep. So we were left. So I